Welcome back to the Learning Studio. This is the Biological Sciences series, and today we're exploring one of the greatest scientific milestones of our time, the Human Genome Project and Genomic Medicine. If you missed earlier genetics and genomics topics like genome editing or population genetics, the full playlist is linked below in the description. Here's a question. What if you could hold a complete instruction manual for building a human being? Wouldn't that change everything we know about health and disease? That's exactly what the Human Genome Project achieved, a blueprint of life itself. The Human Genome Project, a global quest. Launched in 1990 and completed in 2003, the Human Genome Project, HGP, was an international collaboration involving scientists from the US, Europe, Japan, China, and beyond. Its mission was simple but ambitious: to map all 3 billion DNA base pairs in the human genome and identify every human gene. Key achievements included sequencing the entire human genome, identifying around 25,000 human genes, providing a reference map for studying genetic diseases. It was one of the largest and most expensive projects in science, costing about $3 billion, but it changed biology forever. Why was it so important? Before the HGP, scientists studied genes one by one. After the HGP, we had the entire library of human DNA available. This project opened doors to understanding genetic causes of inherited disorders, evolutionary relationships between humans and other species, variations in DNA that explain differences in health, traits, and disease risk. The Human Genome Project wasn't just about discovery, it was about application. Enter genomic medicine. Genomic medicine uses information from a person's DNA to predict disease risk like breast cancer mutations in BRCA genes, diagnose conditions earlier and more accurately, develop targeted therapies such as precision cancer treatments, guide personalized medicine where treatments are tailored to your genetic profile. For example, in oncology, genomic sequencing can identify mutations driving tumor growth, allowing doctors to select the right drug. In pharmacogenomics, doctors can predict how well a patient will respond to a drug based on their genetic makeup. Case studies and examples. Case study 1. Cystic fibrosis. The HGP helped identify mutations in the CFTR gene, leading to therapies targeting the root cause. Case study 2. Cancer genomics. Drugs like imatinib, Gleevec, target specific genetic mutations in leukemia cells. Case study 3. COVID-19. Rapid sequencing of the SARS-CoV-2 genome was made possible by techniques developed after the HGP. Past, present, and future relevance. In the past, the HGP was a monumental scientific achievement, comparable to the moon landing. In the present, genomic medicine is already transforming how we diagnose and treat diseases. In the future, advances in AI and genome editing may allow true personalized healthcare, gene-based cures, and even prevention of many genetic disorders. Why watch this playlist? This playlist covers the 30 most important topics in biological sciences, explained step by step with stories, case studies, and real-world applications. Whether you're a student, teacher, or just a curious learner, this series is your roadmap to understanding life at the molecular and genetic level. So that's our introduction to the Human Genome Project and Genomic Medicine, the project that gave us the blueprint of life and the tools to transform healthcare. In our next lecture, we'll shift to evolution with natural selection and adaptation, the process Darwin first described and the force that continues to shape all living things today. Welcome again to the Learning Studio. Don't forget to check the playlist link below and subscribe so you won't miss the upcoming journey through genetics, genomics, and beyond. Because once you understand the genome, you begin to understand the story written inside every cell of your body.